But just
Feel free.
We got 50 minutes, full time to
right. You're a very good timekeeper, right? I'm not.
No.
No.
Okay.
All right, you're right up front. Keep an eye on the clock. Okay. So what I have today is I'm going to talk about a research project that had a genetics course. You probably heard someone throw out the term forward genetics, first genetics. That's kind of jargon that we use in the field. This you can see is a forward genetics. What that means is we have a phenotype that we're interested in, and we want to understand the genes that contribute to that phenotype. Okay, so in genetics, we're always thinking phenotype. So phenotype is what you see, right? What's expressed, and then genotype is that hidden, but not so hidden thing now because we have genome sequences, references, we have genetic markers, so we can genotype things and understand what's going on in the genome and how does it affect traits. I understand I have a captive audience, so I'm glad Dr. Alba doing it. Finally, but in addition to that, I've adapted my slides to a lot of teaching. Sometimes I get to talk right to all people who know what I'm talking about, so I don't have to do any teaching, but I've got some teaching slides in here for the learning slides to follow that. But in addition to that, if there's something that, well, that doesn't make any sense or understand, please let me know, stop me, and then we can go back and figure out where the gap and how. Okay, fair? Okay. So, obviously, the talk is going to be about breast cancer. We're going to talk about the genetic component. There is obviously an environmental component to breast cancer. And you know about somatic mutations. So it's changes over time. It's an age-related disease. So as mutations accumulate over time in tissue, a cancer program, genetic program develops that way. Okay, so that would be an environmental component. What I want to talk about today is the genetic component. So that's hidden over here in genotype. And so we call these complex diseases. Think of any common disease. Most of them, we're going to classify them as complex diseases. Heart disease, diabetes. Can you think of any more? Hypertension, maybe. So these are all controlled by genetics and environment. Okay? So think about that. And what I'm talking about today is the inherited genetic component. So with cancer, we have to talk about somatic changes, so changes in the cell that's going to become a cancer cell over time. Plus, now we're talking about what's inherited, so the germline variants. So in the germline, the estimates of heritability, so that's the amount of genetic variation that's genetic that contributes to the disease. That's what we call heritability. Okay? And the estimates for breast cancer is about 30 to 40%. And this comes from twin studies where they study concordance in monozygotic twins and compare it to concordance for the disease in dizygotic twins. So if you have a higher concordance in monozygotic twins than you do in dizygotic twins, so monozygotic twins share their genetics. So you would say, well, there's a genetic component to it. And then you can do some regression analysis and figure out how much of the variation is different between monozygotic and dizygotic twins. And when we get into these genotypes, what we can see mostly for breast cancer is that we have a lot of rare inherited mutations. So these are at the population level, they're rare. So they exist below 1% in the population, but they have a high genetic penetrance towards developing disease. So in this categories, think of gene mutations like BRCA1, BRCA2. I'm sure you've heard of them. Down here is where we're going to be mostly studying today. It's these common genetic variants. These are genetic variants that exist at an appreciable level in the population, and they don't have a major effect because of that, but altogether, their effect adds up to a majority of this heritability in breast cancer risk. So what have genetic linkage or association studies taught us? They've shown us that there are multiple susceptibility alleles in the human genome, and there are genetically determined mechanisms include DNA repair. And then where I'm going today is much of this common genetic variation is not well understood at the mechanistic level. We're starting to be able to identify it, but once we get to the point of identification, the mechanisms tend to be unknown. Okay? So the gap in knowledge here is what are the genes that are being controlled by this genetic variation, and what are the mechanisms that contribute to breast cancer susceptibility. And what that makes this so difficult is that a majority of the variants that associate with risk are located in what we call non-protein coding DNA. So if you have had a genetics course, you understand that there's coding DNA, so that's exons. Okay? So if you change an amino acid or introduce a stop codon, that's a little more, that's easier to understand biologically what the pathogenicity is. But if it's outside of the exonic DNA, then it's probably in a regulatory element if it's causal, and it's a little more difficult to understand what genes that variation is regulating, or even how it's regulated. Okay, and you'll see how this problem gets funneled down smaller, and then it gets, as we learn more, then it gets to be bigger gaps in knowledge and becomes kind of big bogs down. So where I come in with my work is I use a rat model to study this. Okay? So this is the, what I'm showing you here is fairly, does this show up, or do I have to switch to a laser? Is my pointer showing up? Okay. So what I've got here is, this is at least current as of a few years ago. There's a few more on here, but this is what we call a lollipop diagram. And it's human genome organized obviously in chromosomes, so that we call this an ideogram. And each lollipop is a location that has been associated with breast cancer. And these red ones are the high penetrance, but rare in the population. And then the blue ones are all these non-coding variants I'm talking about that are basically dispersed across the human genome. So these are the locations of interest. And over here on this graph, this is a little older, but I like how it displays things. It gives you a general idea of what's going on in the population. So here on the x-axis, we have risk or allele frequency. So this would be in the general population. And then up here on the vertical, we have the relative risk. And so as you go up, the variant becomes more genetically penetrant, so higher risk, and then lower risk down here to inheriting a variant. Okay? And you can see this is 1% population frequency. If you draw a line here, these are all genes that were discovered using family studies, because the mutations that predispose to developing cancer are rare in the population. So families need to be studied where the incidence of the disease is higher. And what they found in those studies is that there are, for breast cancer, the architecture is, yes, there are quite a few of these highly penetrant to moderately penetrant mutations segregating in the population. But if you draw your attention to this plot over here, as you increase in allele frequency, what we found with genome-wide association studies or association is that you have all these lower relative risk alleles, and you can see that this is older, so you would put, you know, over 600 dots over here now. Okay, and there's probably been a few more identified since 2022. Okay, so this is where most of the action in the population is happening genetically, and these are variants that if we genotyped us in the room, we would all have one variant or the other. Okay, so if you've heard about the company that went bankrupt, 23andMe, it's a direct consumer sequencing, the genotyping company. They call themselves sequencing, but all they're doing is genotyping, and people send in their sample, they extract the DNA, they genotype it, and these are the kinds of things that we're going to be looking at to come up with a relative risk. Okay? So over here, it's all the variants, whether it's up or down, increased risk, decreased risk, it's all additive over all of the alleles. And these are all variants that one of us would have one copy of, or two copies of, or zero copies of, okay, depending on our genotype. Okay, so I promise you some comparative genomics. So what I use is the rat as a model for female breast cancer. So here's our ideogram of the human genome, and there's a lot of information over here, but what this is showing you the human genome, all the chromosomes here on the x-axis, and then it's lining up or aligning the rat and mouse genomes to each of those chromosomes. So point of all this information is that there's a high degree of conservation. It's just mixed up over the different genomes on the chromosomes, right? So you can see, you're recording this, so you can come back and look at this later if you're interested, or go to this paper where this is a rat reference genome, and you can look at how this works. But here are the chromosomes colored, and that would apply for each of the three species. And then you have the human centric graph, the rat centric graph, and then the mouse centric graph. So here's an evolutionary tree showing just rodents and then human species here. So there is, obviously, we have all mammals. So what makes mouse and rat good for studying female breast cancers? They're mammals, so they all have mammary glands, right? So they all have the tissue that's susceptible. Here, you have some histology images. So on the left here, you can see normal or non-disease tissue, and it showing you human breast tissue and rat mammary gland. So when we talk about human tissue, we talk about breast tissue, breast cancer. When we talk about mice or rats, we talk about mammary glands and mammary cancer, okay? Mammary carcinomas. So obviously, since rats have mammary glands, they do get cancer, and they're a good model for human or female breast cancer because they have a similar histopathology, right? And even the normal tissue, some of the differences here, human breast tissue is more fibrous, and the adipocytes look a little different. They're not as dispersed, whereas you can see in the rodent gland, not as fibrous, and there's more adipocytes. So those are fat-containing cells spread out. But what's similar is you have these ductal structures in both female breast and in rat mammary gland. And it's within these the cells lining these ducts is where most of the carcinomas will develop, okay? It's cancer cells coming out of these tissues. And now over here, you can see some human breast carcinoma tissue and some rat carcinoma tissue. Here you can kind of still get a little bit of the outline of the duct, but what you have is these cells filling up the duct. And here is so invasive that these cancer cells are just completely taken over, filled up these ducts structures over here, and then they've actually invaded out of that basement membrane. Okay. What else on here? So rats, humans, both get hormonally responsive tumors. Both get tumors of ductal cell origin. These are the cells that I talked about. Rats are susceptible to spontaneous. We can also, in the lab, we can induce them with chemicals, radiation, and oncogenes. We can put activated oncogenes directly into the rat mammary gland, and they will develop tumors. And we have also now learned that genetic variation in mammary cancer susceptibility is overlapping for much between humans and rats. Okay? So a lot of shared genetic variation in the trait of interest here. So again, now I've written mammary cancer susceptibility, some kind of population mean, then it's going to depend on the genotypes and the environmental exposures, and then interactions between these genotypes, and then there's also interactions between genotypes and environment. Another way that rats are a good model, because they model the natural progression of breast carcinogenesis. So what happens with, this is, we're using the rat as an example. You go from normal tissue, some kind of insult, the environment gets going, and then one of the first things that happens is you get this hyperplasia in these terminal end buds of these ducts. So you can see the gross anatomy here, then you can see it at the microscopic level, what's going on. And some of these hyperplasias will go on to develop, and they can become these, what we call these ductal carcinoma in situs. Here you have a contained carcinoma. And then again, some of these will go on to develop invasive ductal carcinomas that I showed you before, and how you can see it again. So this is in a rat. Yeah. And human breast cancer develops along the same line. You get these hyperplasia, these ductal carcinomas, these in situs, and then some of those will develop into. This whole process is not understood very well in humans, so the rat becomes a good model to study that, because this gets very difficult. One of the things that animal research does a good job of complementing human studies, right? Because our goal at the end here is to understand female breast cancer and find better ways to predict it, diagnose it, and then hopefully treat it. All right. But like I said, it's a rat-centric talk. And rats have this overlap in genetics, so they differ. We can take inbred strains. So an inbred strain means that it's been mated brother and sister for many, many generations. So its whole genome is uniform. Okay? It's fixed. So it's a diploid organism, whereas most of our genomes, right, we're going to be heterozygous at most loci. A lot of us will be homozygous at some, homozygous for the other. But for rats, if you take an inbred strain, they're homozygous across the board. Okay? And what happened was, way back when, they just fixed a bunch of these rat genomes. There's still variation in the rat genome, but it gets fixed. So if you look at these different strains, like I said, you can induce mammary carcinomas by giving a chemical carcinogen. And you can see different lines for strains are developing more or less or even none, zero mammary carcinomas over time. Okay? So here we have the strain we call Worcester Firth. It's inbred. So its genome is homozygous across the board. And here we have a couple strains down here, Copenhagen and Worcester Kyoto, and they are completely resistant to developing mammary cancer. And this is the polycyclic aromatic hydrocarbon. It's synthetic. It would be a mimic of things you'd find out in the environment of incomplete combustion. Have you ever heard of benzoapyrene? That would be this would be the lab equivalent. So benzoapyrene is going to be in diesel exhaust. Where else? Cigarette smoke. Okay, so this concept's important to understand. We have inbred strains and they have different phenotypes. Okay? So we're going to take advantage of that by crossing them and mixing up their genetics. So we can do these controlled breedings. Here's that Worcester Kyoto strain, doesn't get any tumors. Here's this Worcester first strain that's susceptible. Make a cross, right? This is similar to what Mendel did when he crossed pure lines of peas, right? And he saw yellow and green peas. So he called that an F1. So this is the familial generation 1, the hybrid. So it's 100% heterozygous across the board, right? For the Worcester first, has one chromosome from the Worcester first and the other chromosome from the Worcester Kyoto. And you can see you get a little different distribution from both the parental strains. So this is a first of one of the hallmarks that says, well, there must be genetic variants that are fixed in both of these strains that are controlling the difference between these two. Okay? Then you can take these F1s, in this case, they're crossed back to the susceptible strain, and you can see the distribution of tumors here. So this is the number of tumors that each rat is getting. Rats have 12 mammary glands, so they have lots of glands that are susceptible to getting tumors, right? Mice have 10, humans have 2, right? So you can get multiple tumors, and then if you have markers for this, you can take these animals, genotype them across the genome, and find regions that are being inherited from this resistant strain down here, and find peaks where if the Worcester Kyoto genome has been inherited down here, well, these animals get less tumors, and you get a peak there. So in this cross, they would have looked at multiple meiotic events, and across the averages, they said, well, here are some peaks. We call this a quantitative trait locus, or so these would be loci across here, and across the x-axis here are markers that are spread across the rat genome in this case. And that obviously not as many rat genomes have been studied like human genomes, but enough of these have been studied that where I have these red boxes in the rat genome, this is locations that have been identified. So not quite as spattered as the lollipop diagram I showed you in the beginning for the human genome, and probably the main reason for that is because not as many rat genomes have been mapped for these traits. Okay, whereas hundreds of thousands of human genomes have been used to map that lollipop diagram I showed you. Okay, so how do we go from understanding these peaks? One problem with this study is the genomic resolution is not very good. So we have to narrow these down to get them into syntenic regions. So we're going to try to go back to our susceptible resistant strains. We're going to call this one the recipient, this one the donor. The goal here is to isolate part of this donor strain through breeding of the rats, controlled breeding, so we can select for a certain piece or segment from the resistant donor strain. So again, we cross our inbred strains, we get our F1 hybrid, Now we're going to back cross back to the susceptible strain. So what we're doing is bringing in more susceptible alleles and getting rid of alleles or variants from the resistant strain. Okay? And then eventually we do this enough generations. We've done that. We've selected for example, here, this piece. Now we can come in and inbreed these, and we'll get all three genotypes here. So what we've done is introgressed part of this rat's genome into this susceptible strain. So the idea is, how much does this little segment here change the susceptibility phenotype of this strain? Okay? And the whole idea is there must be some type of sequence differences between here. And it's these sequence differences that are contributing to the phenotypic difference. Okay. And then we go back to our polycyclic aromatic hydrocarbon. We're going to induce mammary carcinogenesis with that. And then at the end of the day here, at 15 weeks of days, we can count the mammary tumors that develop, right? And then we can compare the different genetic strains. Okay. So now, I told you we have to do this over different strains. So what you're looking at here are congenics, and they have different segments from that resistant strain introgressed into the susceptible genome. So different segments. And here are some examples of the markers that we can use to tell where we are in the rat genome. And now you're looking at the mammary cancer susceptibility phenotypes over here, whether or not, so for example, if you look at, let's take N3, we're going to talk about that. This has the resistant allele from the strain from here to there. And if that's inherited or put into the susceptible background, those animals, you get 2 copies of that. So homozygous, they get 3.5 tumors on average per rat, compared to not having this, being the parental susceptible strain, they get about on average 8 tumors per rat. Okay? You do your stats, significant difference, right? And then, so this would be a longer segment of the Copenhagen strain into the susceptible strain, and this is a shorter segment. So this is how we narrow it down. We go from this, somewhere in here, to, well, now we know this, we don't have to worry about this anymore. It's somewhere actually in this overlapping piece. And then if you take all these other strains, this is ruling out independent effects. So these strains have Worcester or Copenhagen, sorry, from here to here, from here to here, from here to here. And you look at their phenotypes, they are not different than the susceptible. So this is how we narrow these down. It's quite labor intensive. It takes some time, but this is how we have to get the resolution down into a smaller and smaller piece of the rat genome. Make sense? A little bit? So once we do that, we get to a piece where we're kind of synthetic between humans. We've got shared synteny between rats and humans. Then we can start doing some studies. And one of the things we can do, I said we can complement studies, human studies with the rat. This is something you couldn't do in humans, but we can do it in rats. We can take donor mammary glands and place them into the interscapular fat pad of recipient rats, and an ectopic mammary gland will develop there. Okay. And this is what they look like. And what we can now do is we can use different donor strains and recipient strains and do reciprocal grafting. And here we have our susceptible mammary glands genotypes into the susceptible recipient. And you can see what the outcome here, about a little over half of those develop. The outcome is tumors. We take our resistant strain and put it into the resistant recipient, and the outcome is about 20% or 25. So we're getting about a 50% reduction, which is what we saw with the endogenous mammary glands. Now these are our unknowns, where we're putting the susceptible mammary gland into the resistant host. and it's still susceptible. So this means the host isn't controlling the susceptibility. But if we take the resistant mammary gland and put it into the susceptible host, we are transferring in that mammary gland, it's something inherent already developed in those mammary cells that are transferring the resistance. Okay? So these are all different reciprocal donor recipients, mammary tissue, and we're growing these ectopic glands and subjecting them to carcinogenesis and looking for different outcomes. This tells us some of these QTLs that we've narrowed down are actually controlled by the host. And when we go do our forward genetics and find out where those are at, it turns out that the immune system is doing something to come into the mammary gland and control the susceptibility. But in this case, we're going to say it's already something in those epithelial cells after they've developed that is causing them to be less susceptible to carcinogenesis. Okay, ultimately, we were interested in the human, right? So if we look at this piece and we go to the human genome and look for the orthologous, we find on human chromosome 5. So over here, we're on rat chromosome 2. Now we're going to jump to human chromosome 5 and look what we have here. There is a breast cancer risk locus there. So there's in a genome-wide association study, they've identified this region as controlling susceptibility in females. Okay, a GWAS usually finds like one SNP or variant that's associated, but in this block, because of the way the human genome is structured in blocks, and there's going to be different variants that develop over time with this. There's a lot more genetic variation in the human genome than there is in an inbred rat, right? So if you look along this, there would be hundreds of other variants in this, but they only tested the one. So what they have to do is figure out, well, which ones are also being inherited along with this, okay? So one of the problems with the human studies is you get one SNP that associates, but all that really does is nominate a region, that identifies the region, so it narrows it down pretty good to the region of the genome. But the problem is there'll be all kinds of other variants in the human genome that are traveling along with that variant that tested positive. So you don't know which one is really causal, looking at a human study. For example, here is the variant that tested positive in that association study. And then they went and looked in many, many more human genomes, and they found that, well, there's all these other variants in this haplotype block, in this population. So they had to test all of them for an association with breast cancer. And when they did that, they kind of divided them up into 3 different kinds of independent susceptibility. But what they found is that across this 250 kilobases, there's at least 193 risk-associated variants that now become candidates for being the causal variant. So where do you think these variants are located? What type of DNA do you think they're in? All of these are in non-coding DNA. Okay? None of them are in exons. They're in introns of these genes. So if you're familiar with looking at a genome browser or that, what you have here are the genes in this region that are transcribed. Looks like there are 3 of them in the human genome. And the big chunks are exons. So these are going to be your most code for amino acids and proteins. And then intervening in the exons, you have introns. Right? These are cut out. And then you have all this intergenic region in between these. So these become the GWAS nominated genes. And then you have all of these potential variants that could be the causal. So the hypothesis becomes, well, one or a subset of these variants are driving expression of these genes. And now where do you go look for that? It's hard to know in the human, right? Because human breast develops after birth, right? It develops in whatever the environment is, and it's also controlled by things outside the human breast, right? It's controlled by hormones that come from other ways. It's controlled by the immune system. But some of the things you can do expression in humans, because you can at least get normal breast tissue and tumor breast tissue, and then this metastatic tissue. So this is from a TNM plot, which is publicly available. And what you can do there is compare normal tissue to tumor tissue. So this is breast tissue in both places. And we're looking at this MIR3 and this MAP3K1. So these were the GWAS nominated genes. All right. And you can see that they are significantly increased in the tumor tissue. And then they go up even further in metastatic tissue. So it seemed to be correlated with progression of the tumor, right? And then at least MIR3 looks like it might be lower or higher expression of that may be associated with survival. So poorer survival with higher expression of this MIR3 gene. Not so much of a difference with MAP3K1. So this MAP3K1 is a serine kinase. It's already been established pretty well that somatic mutations in this will drive breast cancer in about 15% of female patients. Then that other gene here, the SETD9 I showed you, that is not conserved between humans and rodents. So now it's not a good one for us to study in rodents because it's not there. It exists in humans, but not rats or mice. And then we have this MIR3 gene, which was fairly uncharacterized at the time. We've done a little more characterization with it, and we saw this expression. So this is the gene we focused on, this MIR3. So let's look at that. Like in humans, in the rats, we see higher expression of the MIR3 gene in mammary carcinomas compared to the mammary gland. When this is the human haplotype block associated with female breast cancer, this would become the orthologous region in rats, and we can see we have MIR3 and MAP3K1 in here. We look at their expression in the rat mammary gland, and then we incorporate genotype into that. We see that the susceptible strain is expressing higher levels of both MIR3 and MAP3K1 compared to the resistant genotype. So this is the genotype that confers resistance. This is the genotype that confers susceptibility. So the summary of the rat MCS1B and the human 5Q, so they're concordant orthologs. So we've got the rat here down to 1.8 megabases. The one rat strain decreases risk, and we've identified these MIR3 and MAP3K1 as priority candidates in the rat. We look at human 5Q11, so the ortholog, that's a 250 kb region that we're going to worry about. But there are 200 genetic variants associated with female breast cancer risk. Here, the minor allele, so this is the allele frequency that exists lower in the human population, is going to increase risk. And the candidate genes here are, again, there are 2 shared with the rat and 1 additional one. I don't have any data on this, but both of these alleles have effects on the human one. It was found to have an effect on stature, so height, in females, and the rat one has an effect on body mass. So pleiotropic effects here. So if we got 200 GWAS nominated variants in human, are there going to be that many in the rat? So one way we can do that is we're going to sequence both alleles in our resistant and our susceptible strains and find out. So we did that a while ago with some older technology called 454 sequencing. And what we found was there were quite a few variants on the edge of where we thought the rat QTL is, but within the center here, in this orthologous region with the human susceptibility allele, we have 3 variants between the rats. So now we've narrowed this down in the rat from 1.8 megabases to 3 candidate nucleotides. Okay, so in the human, they went from 250 kV down to approximately 200 candidate nucleotides. So this is one of the strengths of the rat, but then this becomes a weakness again between comparing, because these are not well conserved between these sites are not well conserved between rats and humans. One of these, this 17, is on the fuzzy edge of a conserved piece. So maybe within that region, there's some plasticity to play with the genome, okay? Or regulatory effects. So now we can do some functional studies on those variants in the rat. So what we do is clone them into a expression vector. So this is a vector that is controlled by a promoter, and we can put these variants, the susceptible variant, relay associated, and resistant associated variant, and then measure this reporter gene that's called luciferase. It's light, you can sense it with light. This is the same thing, the same gene that fireflies have. Seen a firefly and they glow at night. This is the gene they're expressing of luciferase. So we put this in. We're going to transfect this plasmid into different cells. And these are different human breast cancer cell lines. And then we want to measure the outcome. So a lot of data here. In the interest of time, let's focus on the T47D. So this is an estrogen receptor positive cell line. And we'll look at this 17, where we put the COPE variant in and we get less expression than the susceptible variant. And this is significantly different. The rest of these are kind of across the board, even though they're significant. They don't agree with what we see endogenously with the variants as far as what they're doing with the expression. So the endogenous variants in the rat mammary gland are the resistance is decreasing the expression of the nominated genes. So here we're getting the same effect with this SNP. And we see it in the other cell line. Okay. So let's see where this one is. This is 17. It's in this intron of MAP3K1. These 2 genes, MAP3K1 and MIR3, seem to be regulated together, right? They're kind of like one goes up, the other one goes up with the genotype. So we did something called chromatin, or it's actually chromosome conformation capture, and you'll see it called chromatin. What this is you can test elements to see if they're far apart in a linear distance on the DNA. This is a test to see it. Well, maybe they loop over and they're actually coming together closely in 3 dimensions. So this is what we do. How this works is you have to cross-link. So there's proteins that drive this, that interact with DNA sequences. So it's a protein DNA interaction. You cross-link the proteins that are attached to the DNA, and then you use enzymes that cut the DNA. So now you've shortened this piece up. You get it religated together. Then you do a PCR, and then you compare to different regions in this process so you can detect these things. So how have we done this? Here are the sites. This is where we put a fixed primer. So down here, we would fix this position, and then we test it with different viewpoints. And each of these letters represent a primer that we used in a PCR or separate PCR reaction as a viewpoint. And then you're going to follow this. So now B is here and M is over here. So as you go here, you get farther and farther away from your viewpoint. And you would expect things that are close together would communicate with each other. You're going to pick those up. And then you expect this to drop off. And then if something, so this isn't a huge signal, but it's enough. And we've used all mammary epithelial cells in this, so it's a diverse population. But we are picking up this peak, and this is where these primers would be. So in a sense, these SNPs do come in three-dimensional shape, at least close enough to where they could be. They're definitely close enough to be regulating MAP3K1, but they could also be close enough where they can have an effect on the transcription of this MIR3 gene. Okay? So like I said, the MAP3K1 is pretty well established as a breast cancer gene, whereas not much is known about MIR3, so we're kind of focusing there. And what we've seen is we're looking at expression, and the expression in the mammary gland of this gene is dependent on development. So it's high early on in development of the mammary gland, embryonic, neonate, pre-puberty, then puberty. These, and then it drops down in the adult and comes back a little bit during pregnancy and post-involution. Then we saw in the cancer cells, right, that it comes back again. And that's looking at cancer over here. So you got non-disease, hyperplasia, and all the way to invasive ductal carcinoma. So what this tells us is that the expression of this gene is actually happening highest during when the mammary gland and both the human breast and the rat mammary gland are highly susceptible to things going on in the environment, right? So it's exposures and things during this time, which you've maybe heard of susceptibility windows. These are when they happen, okay? And then we've looked at a few other things as far as expression. So it's depending on where in the tissue we looked. It's higher in the ducts. Those are the target cells. And then it increases with environmental exposure. So we've knocked it out. So we've used CRISPR-Cas9. This is, now we're doing some engineering, right? So this is engineering the rat genome to not express this MIR3. So we've done this whole body knockout. And what we see is that when we knock that gene out, both copies of it, or at least mutate it, we see decreased susceptibility. So our hypothesis is that the MIR3 gene is expression of it at a certain level is contributing to susceptibility. If we can get to the expression level down, those are going to be less susceptible to developing breast and mammary cancer. And look at, I told you that the QTL associates with body mass. So we're seeing the same pleiotropic effect when we knock it out. So this is 2 copies of the mutation, and both females in red and males in green have lower body mass. So weighing a rat is a lot easier than trying to figure out how tall it is. And again, these are like very small effects. So we have to measure a lot of individuals to see this. Okay. So here is a summary, right? So we've talked about expression of the gene, the mapping of the variants. All this supports that both MAP3K1 and MIR3 are genes, but we think MIR3 might be more of a priority in the rat as a susceptibility gene. And then we've identified these variants. So there's quite a few variants, fewer variants in the rat that we can study in the future. And then we've identified this one, this SNP, as a priority quantitative trait nucleotide. Future directions, we want to look at the molecular function of the gene and its role in carcinogenesis, obviously. It's a not very well characterized gene. And then we want to look at the effect of this targeted mutation on gene expression, tissue morphology, and carcinogenesis. So we've gone in that direction a little bit. So these are histology, and I'll just quickly show you that this is the reference, and this would be the targeted mutation. And we don't know what it means. We can only speculate, but we see less eosin staining in these mammary glands. So eosin is the pink color. You can see more pink over here. You can probably visually see this. And there's less of it in the mutated mammary glands. And the eosin stains proteins, not specifically. It likes charged proteins. And a major protein, one thing we thought is that maybe it's having something to do with the extracellular matrix. And that is made up primarily of collagen in the mammary gland. So I've collaborated with somebody in the bioengineering department called Dr. Julia Ebersol. So she has helped me do some electron microscopy. And this is very early and preliminary, but this is the normal non-diseased reference gland. You can see these are the collagen fibrils, how nice and straight, organized they are. But if we start looking in the double targeted mutation, so the homozygous, we see less disorganization, maybe a little shorter. We haven't quantitated a lot of this yet, but we're definitely seeing some visual differences in collagen fibrils. So whether or not these are collagen it has to grow, the fibrils grow in a straight line, and then they're held together with integrins and different other proteins. So we may be seeing some effects on at least the morphology coming through the extracellular matrix. Yeah. My acknowledgement slide. Right. A lot of people help me with this work, and I appreciate all of them. But I didn't leave a lot of time for questions. You can always email me if you don't want to ask them now. But yeah, I don't need to go anywhere. Yes.
How many generations take you to get out to where you can study it? You're breeding and doing the selective.
Yeah, so the standard to make those condense are 10 generations of back crossing is considered standard in the field. You could do something called speed congenetics and test for markers between the two strains and maybe get there in 8. But standard in the field for the congenetics. For these knockouts, we can do a CRISPR. The standard is the back cross away from any off targets, unknown off targets, about 4 generations, then we start doing stuff. And the other thing you can do there, what we do is we use the litter mates as controls that don't inherit any of the mutations or the targeted mutations. Use those as controls as well.
I was surprised at the early slide that the genetic portion is only 30 to 40%. I don't know, I kind of feel like that should be like mostly genetic and then it's triggered by external, but it seems like.
I think maybe where you can help yourself there is when you think of cancer it is almost eventually 100% genetic, right? Because you got that environment and what the environment is doing a lot of times manipulating the genome, right? The those are not genetic. So the 30 to 40%, the risk that's in there. And then that's an average, so it's going to be higher or lower distribution. The highest cancer trait is prostate cancer. That's about 50%, which is in line with heart disease, cardiovascular disease. The heritability is around 50, whereas other traits like stature, the heritability is high there. That's about 90. And using a GWAS for height, I think I'm gonna probably get this wrong, the order of magnitude. But I think they've identified like 20,000 different sites using. Maybe it's 2,000. I might be an order of magnitude off. But whereas breast cancer, it's we're around 2 to 300 using GWAS. But Yeah. Yeah, he used to be at the U of L and then he went to Kentucky 10 years ago now. Have you had him in here to talk?
No, but I won't.
Yeah, you should get that. He is. Yeah, yeah, his work is good. He does work in different like hybrid animal sequencing. He's got a lot of cool stuff. And he's very energetic. You thought I was energetic?
Well, thank you very much for taking the time.
Yeah. Thank you.
Friday. General statement that there's no more demands, there's none. I got the 16th of January, which after the sale again. Everyone have a good Thanksgiving, the holidays, the break.
Hey, how you doing?
Good.
Question. So, like, does the difference in the memory glands that affect the rate at which it happens? You know what I mean? Because like rats, they have 12%. Like humans, we have two. I mean,
Yeah, so I think the way to think about that is they're independent. So see each gland, so rats have 12 memory glands at risk and humans have 2. But that said, does seem to be a difference, not so much in humans, right? Because they're bilateral. But there is a difference between humans in the density, right? And obviously the size of the tissue because you got more at risk, and the density seems to have an effect as well. Whereas rats, yes, the location seems to maybe some of them are a little more susceptible than others. One because some of them are have more cells in them, they're bigger, and the other one is that it might be easier for some of the mammary glands to develop a new blood supply.
okay,
And say more room to develop tumors.
okay, that's
They're
good.
not as constricted. But that whole area is really, it's kind of anecdotal, right? So it hasn't been studied.
Okay.
Right.
And then another question. Now that you mentioned the difference in density, so like the females with a bigger breast are more, what do you call it?
Not necessarily bigger, right? It's just the more dense one.
The
Yeah.
more dense they are,
Yeah,
the more
more
susceptible they are. Kind
yeah.
of. Okay.
Pretty well established. So like the engineering field, that's a good area to go is they're trying to use AI imaging to pick out things that can be better predictors of which images are more likely to develop breast cancer in the future, right?
Oh.
Yeah.
Okay.
Because that's a whole area of prognosis, just trying to figure out why does the normal gland go, some of it stops, right? And then, but some of it keeps progressing.
Okay. Yeah.
Yeah, so that is a big area in engineering with using AI and image analysis,
Mhm.
right? And there's a lot of data out there because
I bet there is, yeah.
women get their mammograms.
Mhm.
All those data are being used to try to train
The AI system
into identify lesions and things that are
Mhm.
going to progress.
Okay. Yeah, that's good. Hey, thank you. Okay, you have
You
a
bet.
nice day.
Yeah, happy Friday. I'll let you come over here. You probably got to stop a recording. All right, thank you. What you're looking for? or-
Yeah.
Yeah.
I like it just because I think people sometimes forget